Hello, everyone. Thanks for the kind introduction and the great lead-in. Again, my name is Alison Manchester, and I'm really grateful to be here. My first time in South America and first time presenting at the Cannes Summit. So let's just get into it, since we only have a few more lectures until our wine tasting. I just wanted to give a really brief introduction to what I'm going to be talking about this afternoon, because we've had many hours of introduction so far, and many of the themes that have been discussed already I'm going to sort of echo, but I'm also hoping to sort of delve deeper into those specifics and the mechanisms. And we're going to do that within the context of chronic enteropathy in dogs. And it's not just because that's my kind of research area of interest, but also because this is a perfect disease, I think, to reflect the themes that are being discussed in these last 2 days of lecture. Chronic enteropathy, for context, for those of us who don't see patients on a daily basis, super common condition in clinical practice in veterinary medicine, both in general practice and in specialty. Turns out to be the #1 cause of chronic diarrhea in dogs. And despite a lot of efforts to better understand this, really a syndrome, we don't understand it very well. And for this reason, we have to take a trial and error approach to management. The good thing is most of our patients will get better if we find the right diet for them. And so again, a perfect disease process in which to consider these themes of immunity, inflammation, and nutrition. So up on the screen here, you can see some of my recent patients I've seen over the past few years, all coming to see me with clinical signs of chronic enteropathy. And what I wanted to stress here is these patients really all kind of look the same to me if I use my typical diagnostics, my typical clinical approach of physical exam, history, blood work, imaging, endoscopy, histopathology. These patients all pretty much look exactly the same. They might have subtle differences in clinical signs, but these dogs are all quite similar. And unfortunately, when they first come to see me, I have no way to predict and tell the owner, give them kind of hope and assurance that their dog is going to get better. And we know that anywhere between 15 to 40% of these dogs are refractory to all of our interventions, despite kind of the wealth of diets that we're really lucky to have in the United States and all of our other interventions. And so this kind of percentage is just a number, but when it turns into your actual patient, it becomes a lot more meaningful. And I wanted to highlight 2 of my recent patients who unfortunately fall into that minority category of non-responders. And the first one is Opal. And you can see she's an Australian Shepherd, and she suffered for many years with chronic mixed bowel diarrhea and intermittent sort of hunger strikes. She obviously didn't feel well during those periods. I think I was the 3rd internist she came to see during her long course of disease. She failed all the diets we could think of: steroids, enteral nutrition, antibiotics, cholestyramine, fecal transplant. Continued to struggle with her disease to the point where her owners felt her quality of life was really too poor, and she was euthanized for her disease at age 6. My other patient is still with us. This is Bruno. I actually just got a text from his other internist about him yesterday afternoon. He also suffers from horrible chronic diarrhea. He's lost a ton of weight, also has failed all the usual suspects in terms of treatments. You might see, is a great Pyrenees mix, and he's 25 kilograms, so something very wrong going on. And I will say both of these dogs had a complete workup, everything we could do at Colorado State, and he continues to have diarrhea. So it's really patients like these that continue to motivate me to take new and different approaches to these disease processes so that we can give them better answers and improve their outcomes. Luckily, I think we're in a really cool era where we have access to a lot of different tools that allow us to comprehensively and specifically look into these disease processes without the typical limitations we have, because this is a canine disease and we don't have this antibody or this reagent. So this kind of omics era allows us to not kind of be reliant on those reagents and look at things really broadly. The important thing I wanted to point out, however, with this sort of omics approach is that we always need to follow up those initial kind of exploratory studies that cast a really wide net with specific tests to kind of hone into those individual genes or molecules or metabolites and see what they're functionally doing in our patients of interest. And so we really want to put these kind of tactics together to move the kind of ball forward on disease and outcomes. As Dr. Lappin alluded to and really set me up nicely for, we've really been interested at Colorado State recently, been interested in applying transcriptomics or the study of different messenger RNAs to better understand disease. And this just offers such a nice step forward beyond what we could typically do with things like PCR and IHC, where we can really, in a less biased way, look at which genes are kind of dysregulated and differentially expressed in health and disease. And of course, I want to make the point that these mRNA signatures are not necessarily completely reflected on what's going on in the actual patient, because we know there can be different factors that alter an mRNA turning into a protein or a metabolite, but I think it's still a big step forward in terms of where we have been. And we can take this even further and get even more specific by using single-cell RNA sequencing, and I'll address that a bit more as we go forward. You might want a little bit of kind of proof that transcriptomic approach can actually make a difference in understanding of disease, and I'm being a little bit biased here. I myself have celiac disease, so I'm particularly interested in which kind of discoveries and kind of advancements we've made in that disease process with the application of transcriptomics. And you can see here results of a recent study employing bulk RNA sequencing for patients with celiac disease, either as they're just diagnosed or after they've been on a gluten-free diet, like I hope that I am. We'll see what happens in the next few days. But essentially what they were able to find with an unbiased approach is some pathways and some implicated issues that would never have been hypothesized before taking this really broad approach. So they found that even these celiac patients, again, like me, who should be in remission with a gluten-free diet, they still have big alterations in the spliceosome, in their cell adhesion molecules, as well as in disruptions in their epithelial regeneration. And they hypothesized that that could be why some of these celiac patients have refractory disease and continue to show signs despite a strict gluten-free diet. Another group recently used single-cell RNA sequencing just of immune cells in the duodenal mucosa to further investigate this disease, and again, found some unexpected findings. They were able to document a loss of these putatively protective natural intraepithelial lymphocytes in the active celiac patients, and then they also found some less expected myeloid cell differences between patients on gluten-free diet and those still ingesting gluten. And I think these findings provide a lot of motivation to apply this approach to our patients. So our objective with the data I'm going to present today is really to compare the molecular programs and cell populations in endoscopically obtained duodenal biopsies from client-owned dogs with chronic enteropathy to a healthy control population. For our patient populations, this is always a bit of a challenge as what really is healthy when it comes to GI disease. We can't, do a GFR or look at a creatinine level. So I will say we took animals that were fed a consistent diet that showed no clinical signs, that had normal blood work, and compared those to samples obtained from those client-owned dogs, similar to the ones I showed a few slides ago, who had the typical signs of chronic enteropathy with no other kind of documented reason for their signs. And I will just re-emphasize, these are not PLE dogs. These are dogs with normal serum albumin. Kind of outside, they look like they're fine, but they have these refractory clinical signs. For the single-cell arm of the study, we had a smaller population of dogs whose samples we processed for single-cell. That's for a few reasons. First of all, it's a really labor-intensive process to process a duodenal biopsy or a dozen duodenal biopsies for single-cell, and it's also markedly more expensive. So, of course, we live in a world where we have to pay for things, so that's why we had a smaller group for that study. basic overview of how we actually did this. I did an endoscopy on all of these dogs, obtained biopsies like any of us would. Those were just put in RNA later. RNA was extracted and they were sequenced at Novogene. A little more intensive workflow for the single cell study, as we had to take those little grain of rice pieces and digest them to get a single cell suspension. And epithelial cells, which we've heard a lot about today, that barrier, it doesn't actually like to break apart. It likes to stay together in these little clumps. Clumps are very bad for the single cell instrument, that 10X machine, that I really didn't want to break. So we did a lot of enzymatic and physical digestion to get into a nice single cell suspension of live cells. That suspension, we aimed for about 5,000 cells per dog, and that went into that 10X genomics machine, which is this small tabletop machine that uses microfluidics to kind of mix those individual cells with these functionalized gel beads, which allow this kind of, to me, unbelievable chemistry and science to happen in these little gel and bead emulsions, whereby the cells are broken open, the RNAs are released, and they're tagged with individual barcodes so that later on down the road, I can get those transcripts mapped back to the individual cell of their origin. After all of that takes place, a cDNA library is constructed just like with bulk RNA sequencing, sent off again to Novogene, and then we have, I would say, months to maybe a year of data analysis to get where we are today. So in terms of what I wanted to share in our transcriptomic investigation of this disease, we're going to start off with an atlas, because honestly, though we've seen a lot of beautiful kind of pictograms and biorender images of the gut mucosa, those are extrapolated from mouse studies and from people. And honestly, we don't always have this data in dogs, and now we have a little bit more clues and actual data to see if what we think is going on is actually truly going on in our patients. So as an overview to start, we can look at this UMAP plot, where each individual dot represents an individual cell from an individual patient, and you can see here representation of about 35,000 cells that we sequenced from the 7 individual dogs. And we ended up with 7 major cell populations. Not surprisingly, our biggest population were T cells and epithelial cells, huge numbers of those cells, with smaller contributions from myeloid cells, plasma cells, cycling T cells, mast cells, and B cells. How did we come up with those identities? Well, we used some markers which are more kind of widely understood that we would use for things like flow cytometry, like CD45 to highlight immune cells, CD3 to highlight T cells, and then some genes you may not be as familiar with, things like MUC13, which is a mucin gene, which is expressed highly by epithelial cells, KIT, which was expressed more preferentially by mast cells, lysozyme for the myeloid cells, and CD20 for those cycling T cells and plasma cells. The one thing I wanted to point out here is that the genes that were most kind of characteristic of individual cell populations were not always kind of in line with what we expect based on the kind of canonical markers that we typically use and that we have antibodies for. So I think this was an important kind of learning point for me to realize that some of the markers that we really kind of hang our hat on sometimes are not as specific as we might expect, and we should keep this in mind when considering other studies. With single cell, there's a lot of complexity in the data, and we can kind of zoom in further. So when we did that with the epithelial cells, that was pretty fun to be able to see, wow, we have a lot of kind of transcriptomically unique cell subtypes that correlate with things that we've been talking about for decades now. So if you notice here in the middle, we have this more kind of transcriptomically similar group of enterocytes, as well as these interferon-expressing enterocytes. And then more transcriptomically distinct were smaller populations of things like goblet cells, tuft cells, enteroendocrine cells, and epithelial cells expressing bestrophin 4. When we did this for the myeloid cells, we had maybe more recognizable cell subtypes, as we can find these in the peripheral blood, and we found groups of neutrophils and eosinophils, 2 types of dendritic cells, as well as monocytes and macrophages. probably more exciting than just, establishing an atlas is actually, well, what was different between health and disease, and this, of course, was really the motivation behind this study. And again, complex data set, lots of places we could go with this, but to start kind of as a roadmap of what we want to focus on, we can look at simply the number of differentially expressed genes within those different cell subtypes. And you can see here we actually had the largest number of differentially expressed genes within the myeloid cell population, followed by epithelial and mast cell, with many fewer differentially expressed genes as we get down the table. And again, the direction here is chronic enteropathy versus healthy, so 115 upregulated genes in myeloid cells from chronic enteropathy compared to healthy. I did want to point out before we go on, I'm not going to talk anymore about the mast cells, though I think they could be an important cell population in chronic enteropathy. However, I think we only isolated like 400 mast cells total out of the 35,000 cells, so we didn't have enough cells to do further analysis there. But going back to the myeloid cells, again, with a kind of a bird's eye view overview, we can start off on looking at what were those differentially expressed genes, and you can see that here in the scatter plot, where the red genes above that X equals Y line are upregulated in chronic enteropathy versus health, and blue are downregulated. And some of these genes might look somewhat familiar to some of you, things like S100A12, SOD2, and NFK-BIA all might start to give an idea about neutrophil activities and function. However, we can sort of check ourselves and see if that's an accurate interpretation of the data by looking at this split UMAP plot where we highlight some of those genes that were differentially expressed and find out which cells they're coming from. So top pane, healthy, bottom pane, cells coming from chronic enteropathy dogs. And I want to draw your attention to these cells that correspond with the neutrophil population. And you can see these widely known pro-inflammatory genes like IL-1 alpha and beta and superoxide dismutase 2 are really overrepresented in the cells coming from the chronic enteropathy dogs that are absent in healthy. Interestingly enough, we actually found no numerical differences in the number of neutrophils present in those biopsies, health versus disease, which could be related to smaller sample sizes since we had fewer myeloid cells than others. However, the main conclusion we made, at least at this point from this data, is that we have an overactive population of neutrophils in the duodenal mucosa of chronic enteropathy that are absent in those healthy dogs. Next, I wanted to look at differences within the epithelial cell populations, and hopefully I don't have to go into this slide much because I think we've had a lot of good arguments about why we should care about the epithelium. However, I did want to draw your attention to this review article published a few years ago, which I thought really nicely summarized some different studies published in the year 2020 that really further implicated the role of the intestinal epithelium in the pathogenesis of IBDs in humans. And the two studies that I found most provocative were one, finding that increases in intestinal permeability preceded the onset of Crohn's disease in humans by years and really was able to predict the onset of that disease, as well as other studies in rodent models showing that the intestinal epithelial expression of MHC class 2 is actually diurnally regulated and influenced by diet, and that in turn regulates this intraepithelial lymphocyte population that makes IL-10, suggesting a real kind of strong connection of the epithelium, kind of telling the immune system what's going on and helping shape those responses. I think maybe the intestinal epithelium has not necessarily been ignored, but not paid as much attention to as other cellular populations in veterinary medicine, specifically in chronic enteropathy. However, there are older studies, for example, Roger Batt's group looking at intestinal brush border enzyme activities, as well as kind of one of my honestly favorite studies that I have seen coming out of the chronic enteropathy space in the past decade, out of Oliver Garden's group, showing that if we kind of look in the right place using transmission electron microscopy, we're able to see morphological differences in that gut barrier in active disease that are actually resolved when those animals are fed a therapeutic diet and they go into clinical remission. Interestingly, and I wanted to also point out, if you look closely at this study, they actually found no differences in the numbers of lymphocytes and plasma cells in their healthy population versus active disease. So I think, we should really put focus into areas where we have objective evidence that this is potentially contributing to disease. So this is a time when I wanted to kind of draw in results from our bulk RNA sequencing arm of the study. Again, that was 7 chronic enteropathy dogs versus 3 healthy beagles. And interestingly enough here, and again, this is I would say, highly unbiased approach. Our top upregulated genes are really commonly mapping back to epithelial cells. So things like cytochrome P451A1, a gene that encodes a chloride channel, and then an SLC gene that encodes for a sodium channel. And you might say, okay, well, she's just saying that. Are these genes even expressed by epithelial cells? Well, the cool thing is that now that we have this single cell data set, and this should be a publicly available cell browser in the near future once this paper is published, but essentially I can go in and type in any gene that I'm interested in and find out who's expressing it. So if we do that for CYP1A1, you can see here it's almost exclusively expressed by canine duodenal epithelial cells. If we look at that chloride channel, again, highly expressed by epithelial cells. So I don't know about those of you in the audience, maybe this is just me being really in the weeds about my own research, but I think this is a really cool tool to be able to have. And you can come find me afterwards and we can look at whatever gene you're interested in. I don't want to only focus on the genes that are upregulated, because downregulated genes could, of course, be really important for disease pathogenesis. And interestingly enough, again, here are the kind of most significantly and most extremely downregulated genes are also mapping back to epithelial cells. These kind of more specifically seem to be related to stromal cell gene expression, and suggesting that differences in kind of cell structure or tissue structure and extracellular matrix activities seem to be downregulated in the disease population. Another cool thing about single cell is that we can kind of parse out differences that are driven by differences in cellular relative abundances. So I think one interpretation of this data could just be that, there's more of these cells in disease and there's fewer of these cells in the disease, but we can actually disprove that theory by looking at these relative abundance plots, where you can see green dots show the relative abundance contribution from the healthy dog duodenum, and the purple dots represent contributions from the CIE dog duodenum. And apart from tuft cells, where we almost have a significant difference, and maybe I could convince you that there's fewer tuft cells in the chronic enteropathy samples, we essentially have broad overlap and similar contributions of those different cell subtypes in health and disease. So that was kind of nice to, rule that out. It's not just a cell numbers type of thing, which I think further supports the validity of our bulk RNA sequencing data. But so what are those differences that we actually saw? I just highlighted 2 genes that were significantly differentially expressed, health versus disease, and you can see again the split UMAP plot, where if there's a purple dot, that gene is highly expressed from that sample, and you can see here CYP1A1 is highly upregulated in enterocytes from chronic enteropathy, and we have the opposite pattern in this KCNK16 gene, which encodes for a potassium channel, which is almost has a lack of expression in those chronic enteropathy enterocytes. For the purposes of today's talk, and it became even more relevant, I was excited to hear many different lectures that we've heard over the past 2 days have been mentioning this aryl hydrocarbon receptor. And interestingly enough, CYP1A1 is actually a effector molecule of that AHR. And again, just to kind of prove that I'm not just making something up, aryl hydrocarbon receptor is highly expressed in epithelial cells in dogs, but also in myeloid cells and T cells. And work in other species has shown kind of a variety of activities that CYP1A1 appears to be responsible for. You may have noted that the aryl hydrocarbon receptor was first known for its ability to, or its importance in kind of sensing environmental toxins and metabolizing those compounds. So we know that CYP1A1 is important for oxidizing polycyclic aromatic hydrocarbons as well as metabolizing polyunsaturated or fatty acids into other bioactive lipid mediators. But essentially, for the purposes of today, since we're not going to go into a real deep dive in molecular biology, we can just think about CYP1A1 as being a really important negative regulator of the aryl hydrocarbon receptor as it breaks down the ligands that go back and would continue to signal to the AHR. What do we know what might be a consequence or an outcome of increased expression of CYP1A1? Well, again, kind of relying on findings that have been fleshed out in rodent models, we can find that increased expression of CYP1A1 has been associated with oxidative damage and intraepithelial lymphocyte death. And in some models, that's actually been significantly associated with increased susceptibility and death and poor outcomes from enteric infections. And kind of even more interesting to me, especially as I have this interest in what is the role of the intestinal epithelium in these patients, they found that actually excessive expression of CYP1A1 is associated with kind of heightened intestinal epithelial cell proliferation and lack of differentiation, so that these cells can kind of like pile up, and that interferes with normal intestinal epithelial barrier functions. Cool thing is, for the purposes of this conference, is that we know we can kind of rescue that phenotype with the right kind of immune and nutritional intervention. So in mouse models, again, they know that we can feed those aryl hydrocarbon receptor ligands, which include indole compounds and things we get out of cruciferous vegetables, and actually rescue that phenotype whereby the intraepithelial lymphocytes regain their activities and the intestinal epithelial cells differentiate normally. So again, this is a preliminary finding in our initial kind of exploration of health and disease, but to me this is really exciting in a potential area of further exploration to help these patients. Finally, in the intestinal epithelial cells, you may have noticed that those two genes that I highlighted kind of mapped back to this enterocyte 2 population at the bottom. So we were interested in kind of the more global differences in those cells between health and disease. And you can see here the results of our pathway analysis. And essentially, these red bars represent pathways that are upregulated in chronic enteropathy versus health, and blue pathways downregulated. And the text may be a bit small for those of you in the audience, but I will just go ahead and tell you that we seem to have issues with sort of epithelial cell kind of repair and kind of reactions to injuries in the upregulated pathways. And perhaps even more interesting, the downregulated pathways all seem to map back to epithelial cell metabolic processes, suggesting that metabolic kind of defects could be present in the chronic enteropathy samples. For those of you who are interested in chronic enteropathy, you may be sitting there like, why is she talking about the epithelium and myeloid cells? Like, it's T cells, it's always T cells. We're always looking at T cells. Well, you will soon realize why I'm not focusing on them. Honestly, the picture of the T cells was extremely complicated and difficult to sort of make a story out of. But even just starting here, you don't need to be any sort of transcriptomic expert to see we have this sort of blob of cells, and they're all very close together, suggesting that there's a lot of kind of molecular gene expression overlap between these different populations. You may notice some populations that seem more familiar, like natural killer cells or regulatory T cells, but other of them are much more kind of obscure, and we kind of had to look at the gene signatures and assign a name, but honestly, this was really challenging, and I would say the story is much more complex than what we might sort of paint the picture based on samples from peripheral blood mononuclear cells. We also found no difference in relative abundances of different T cell subtypes within our small patient subsets. I did want to highlight regulatory T cells, as There's been kind of conflicting data to suggest if these are more or less present in the duodenal mucosa of chronic enteropathy dogs, and at least from this data set, we found no difference. So at this point, I wanted to just take a brief pause and just admit that this is kind of one approach using transcriptomic analysis of the duodenal mucosa to try to advance the understanding of chronic enteropathy. And of course, there's many other kind of parallel and complementary approaches we could take. And of course, we would need to confirm any of these sort of initial inclinations and see how relevant they are for our actual clinical patients. So I kind of, when I get towards the end of a study, my usual question is, well, what now? Yeah, we have all these data and we have these studies and it seems exciting, but we have to actually do something with them. So like I already alluded to, we really do need to add in these sort of complementary approaches, add in our data from metabolomics. What are the bile acids doing? What are the microbes doing? And see how those work together, because we're obviously not just, working in a sterile system. So we have to ask ourselves which path to take, and this is a path I like to take in Colorado when I have a day off. But some of the questions I'm really interested in are, these differences we found, is that a consequence of the disease because, my gut barrier is upset and I've had all these antibiotics and all these previous medications, or did this actually cause the disease? And we would, of course, need longitudinal samples to answer those questions. I would also kind of be skeptical of my own data and say, are these gene expression differences reflected at the protein level, which would be more physiologically relevant? And of course, relating them to other omics platform results. Besides going forward, I think it's also important to look back and think about how do these results kind of fit in with the data set that we already have and all of the kind of decades of data that have been generated before I even started thinking about chronic enteropathy. And I think one sort of nice proof of concept is that we've had other clues that neutrophils are important in this disease process based on concentrations of fecal S100, A12, as well as some histopath evidence that duodenal neutrophils are important. In terms of the story on the T cell side, I think it's a little bit more complicated. And honestly, I am quite skeptical. You can probably already tell of the role of T cells in the pathogenesis of these dogs' disease. And I think the data we have this far is not extremely strong. One interesting thing I did want to point out as we wrap up is kind of looking at these typical T cell subsets, like we're always saying CD4 versus CD8. I just plotted here for you the CD4 and CD8 expression in our duodenal T cells. You can see we didn't have a great kind of signal from CD4, which could, I would admit, be somewhat due to kind of our analysis technique. However, that didn't seem to be CD4 versus CD8 was not a big differentiating factor in assigning a identity to those T cells. Other kind of classical cytokines that we associate with different T helper subsets also didn't seem to really make any difference. Different markers, though, that we don't always think about, things like granzyme, actually did provide us a better differentiation to split some of those T cell subsets. So honestly, when I first was looking at this data, I was a little bit worried and thinking that, what I got couldn't necessarily be accurate. However, this has actually been pretty well proven in our other species. Here I'm just showing results from a really interesting study that I came across last year, whereby they took mouse and experimentally infected them with different infections that were expected to kind of bias the T helper signature and kind of push the T cells towards a certain response that we would expect based on what we learned in immunology. And what they found actually was the gene sets actually were not reflective or differentiating in terms of the T cells they found in those mice intestines. What was much more differentiating was just the kind of pathogen themselves, suggesting that our sort of canonical T helper 1, Th17 are really perhaps not as physiologically relevant as we might hope they would be, or based on what we've kind of been taught. So I think for me, this is just a bit humbling, but also makes me kind of optimistic for taking these approaches and actually finding out what's physiologically important for these patients. It also makes me think of a line I read in a book last year where the author pointed out that we don't study what is there, we study what we can study. So what do we have the antibodies for? What do we have kind of initial data that suggests this is a pathway to follow? If anyone has, like, I would say 6 hours a day that's just lying around in their schedule, I would highly recommend reading this book. It's long and it's fairly dense, and if I'm being totally honest, I didn't actually finish the whole book, but I'm planning to get it back from the library when it's not on hold. But again, really interesting read, and it provided me with some really nice context, especially as I was writing up my dissertation, to kind of revisit how I'm looking at the results that we have from some of our research. Of course, all of us, especially those of us on the clinic floor, want to know, like, what can I give? How can I modulate the immune system and make it work better for my patients? I would say I'm not really going to answer that question today because I simply don't have an answer that I feel like is valid and true based on the data I have right now. However, I did want to highlight some recent studies I've come across that do kind of show a nice mechanistic link between a molecule and a change in epithelial barrier function. We've talked about that a lot today. We want to strengthen that epithelial barrier, but how do we do that? And at least this group, again, we have to start somewhere. They're starting in a mouse model, but essentially they found that when they supplemented this bioactive lipid, lysophosphatidic acid, they were able to restore barrier function. And when they gave that bioactive lipid but took away this receptor, they actually had worse epithelial permeability, increased bacterial translocation, suggesting that kind of molecules that we really might never have thought of could be really important and impactful for improving intestinal epithelial barrier function. Otherwise, and also going off of our signals that CYP1A1 expression could be important, and knowing that they are able to kind of rescue these intestinal epithelial functions with those aryl hydrocarbon receptor ligands and also antioxidants, I think that would be another area that we really need to further explore, particularly in these patients. And finally, based on that signal implicating myeloid cells and overactive neutrophils being important in these patients, I'm really curious, well, what was the thing that was actually activating and kind of upsetting that neutrophil in the first place, or kind of motivating it to come into the duodenal mucosa from the peripheral blood? And for me, I would much rather kind of nip that in the bud rather than sort of like suppress the neutrophils once they're already there. So a lot of interesting and exciting directions we could go forward with this, and I really am excited to be, doing research in 2024 in this time when we can do some of these really advanced techniques and move beyond the sort of conclusions and findings we can get from histopathology. No offense to pathologists, as I know there's at least one in the room, but yeah, kind of adding a more broad picture by adding in all of these different approaches, trying to intermesh our findings. We've now developed a tissue atlas for the canine duodenum, and I'm just really excited to see what kind of advancements we can make by kind of combining our expertise and finding out what's really there more than just what we hope or thought might be there. So with that, I thank everyone for their attention. I know it's a hard time of day to stay awake, but now we're just an hour closer to our wine tasting, and I'm really happy to take any questions.